Here this early in the morning. This is a very auspicious event. It's the first annual workshop for what hopefully will be the Center for Brain Science that we're starting over here. This particular workshop is going to be on the big data revolution in neuroscience. And so we've had a number of people come down here all the way from Minnesota that are going to help us with that. And I wanted to just at the top go through some preliminaries, do some logistics, and then we'll get into the actual science. So what are the goals of the workshop? Or why are we here? The main scientific goal is to really gain hands-on experience with current neuroimaging methods. And we'll do that particularly for big data sets, very large data sets. And we'll actually get hands-on experience with that because Kendrick Kay, who's over here, together with his collaborators, including Emily Allen, have collected 70 data in a number of observers, but those observers have been very much extensively put through the ringer of the scanner. So they, I don't know how many hours they actually spent in the scanner, but they pretty much lived there. Like they could just live in that scanner room. And we'll get more information on that today on the first day, what actually happened. And this is, of course, ever accelerating, right? So the ability to collect data is growing at a much faster pace than any individual lab can keep up with. And that also means that we're going much more to an open science, open data kind of approach, where you might not necessarily collect the data that you'll be analyzing and that you'll be interrogating. And so that is really what we're going to be doing here. There's a secondary goal that's really important to me, and that is really to get a critical mass of expertise at NYU Abu Dhabi in doing these kinds of questions. And that specifically goes into this context of a Center for Brain Science, which is really pulling together cognitive science or cognitive neuroscience, data science principles, and also something that probably won't be in this workshop, but maybe the one next year, a health science component. So once you actually understand how the brain works, you can apply it to clinical populations, see what goes wrong in that case, and that might help you improve diagnosis, prognosis, and treatment. And that's something that also will be very much a part of that center. So you've already seen me. I'm the workshop organizer. Any questions? Anything that goes wrong, that's on me, and you should let me know. Anything goes right, that's on the other people. So first, there's our instructor, Kendrick Kay. He'll be doing most of the talking, at least this morning. He's at the University of Minnesota, as is Emily Allen, who is one of the teaching assistants. She'll be the primary teaching assistant for the first 4 days of the workshop, when we were talking about functional MRI data. And then Nate, who's sitting right over here, Nate Miller, he will be helping out with the last day, the 5th day, where we're looking at diffusion MRI data. You should also have a handout that has everybody's email address and contact information in case anything comes up. If you don't have a handout, raise your hand, because there should be enough around. Is there a big stack somewhere? There it is. That handout will be useful because it basically has an outline for the program. It will tell you where the data lives. It has our contact information and other logistical information. Any questions at this point? Pretty straightforward, right? We haven't really covered any complicated topics. Okay, so we get to some logistics. The schedule, it's intensive. It's really like this whole week, we're going from 9 to 5. We may not go to 5 every day, but we're doing this for 5 days. And I expect that by the end of it, we won't be tired of imaging data, but we certainly will have had our share for like a little bit. The typical structure that would deviate from that every once in a while is that in the morning we'll have lecture sections, which hopefully will be interactive. You should ask questions at any time when they come up. Don't wait until the end. And there'll be time for lunch. Most of you are from NYU Abu Dhabi, and so you already know about your lunch options, but I'm going to go over some of them for people that are visiting from other institutions. That goes until 1:30. And then the afternoon typically is hands-on, which means that all of us move over to the computer section and we actually get our hands dirty on this data, which I think is a much better way of learning than going through lectures and you can nod along and say, okay, yeah, I completely understand that. But then doing it yourself is usually a different question. If you are not familiar with the lunch options, there's the student cafeteria, which is on the far end of campus over there. There's the marketplace, which is in this building on the second floor, or floor 2, let's call it that. There's sushi counter on that side of the university. Coffee, snacks. There's a cafe right outside the library. There's a convenience store if you go to the floor 1 of this building. And then there's Blacksmith, which has good coffee, but some people will tell me that it's expensive. So you're on your own for lunch. That doesn't mean that you have to go have lunch by yourself. It's just if you want to meet up with people, you should make that. You should take that initiative. We won't have lunch as a group. That's basically what that means. Please keep food and drink out of the computer lab. I know that water bottles are okay, but be smart about it, because if we have a big spill, we get kicked out, and then nobody's happy. Nobody gets to do that work. So water bottles okay. If you have them and disguise them as coffee bottles, probably also okay. But don't bring your lunch in here. Then to conclude, at the end of the whole workshop, we'll have a dinner. That dinner will be at Almayas at the Sheraton. It will be Thursday at 6:30 after the conclusion. Do let me know in the next couple of days if you're going to able to make it, because I need a head count. I need to tell them how many people are going to actually be at the dinner. And I think that's it in terms of the information that I have before we dig in. Any questions at this point? Good.
I was also going to mention, for the few people that don't have access cards, some of the bathrooms will require access cards to use them. So the ones that don't require them, so there's the entrance to the library that you all came in. You go down either one of the sets of stairs, and there are bathrooms around the corner either way.
Yes. That also means for people who have access cards, the closest bathroom is right on the other side of this wall. But it's a weird setup where you can only get in there if you have NYU access. Other things? That's it. Okay, so then without further ado, I'd like to hand it over to Professor K. He'll probably go by Kendrick for this workshop, who is going to start and really getting us set up on what the data, well, first of all, principles of fMRI analysis, and then we'll get into what this data set is that we're going to be working on for the remainder of the workshop. Thanks a lot. Thanks for coming. I'm really excited about this. I'm really happy this is coming together. There's always this question about, what if you throw a party and nobody comes? I'm happy you all came to this party. We'll have a good time, and I'm really looking forward to this being a continuing thing that we'll do every year, and pulling lots of people together from all over the region. Thank you.
Very complicated. So actually, first question, there's a slight latency. Is this annoying? Should I turn off the PA? It helps, but it's slightly disconcerting.
Can you turn it down a little bit maybe, because I think it's for the speaker.
We tried to set this all up, and it's quite complicated. Okay. Is this going? Cool. So I'm Kendrick. I'm going to stand up. And I guess this first bit, the timing of this is difficult. We'll see how long things go, and we might slow things down or speed things up depending on interest. This first bit is really just about why we're doing this and what the goal of this workshop is and how it was designed. It's hard to multitask. Okay, so in no particular order, let me just orient you to the syllabus. Feel free to read it offline, but I just want to highlight one bit of it. So if you look on the back, let's see. So these lecture slides, it is a bit annoying. Maybe we should just cut it off.
Just go down a little bit. I think it'll be helpful.
So all the slides are already made available. They'll be on the computers, these iMacs and the laptops, and there's a Dropbox link. Actually, it's a shortened URL, tiny URL. You can access the slides at that link if you want to, on your own computer if you want to right now. Let me know if it doesn't work. Also, let's see, what else is relevant? Yeah, so this computer stuff will be more relevant in the afternoon when we'll actually sit at these machines and navigate and look at stuff. Oh, and then the last section on the syllabus here is these little student projects. Basically, jumping ahead a little bit, we prepared scripts, example scripts that you don't have to do. You can just run them and they work. But of course, that helps to some degree, but it also helps if you were to actually do something new yourself. And so I just listed here some examples. So over the course of this week, if you find yourself bored with the example scripts and just want to plow ahead and pick one that seems to interest you, we can break off in little groups, just like one of us or me and you or whoever, and we can work on making these scripts. These don't exist, the examples exist. So anyway, so that's what the meaning of those student projects are. You don't have to do one, this is not a class. Okay, and one last thing, there's a Zoom ID on the second to last section at the back. So during the afternoon, when you're sitting at your computers working on your stuff, it's gonna be hard to coordinate, but sometimes we wanna show stuff, or maybe you wanna show me what's happening, or you wanna show everyone what you're facing. If you zoom in to this call from your iMac, you can then share your screen, and then it'll show up on this gigantic thing, TV. Anyway, so that's the Zoom ID that you'll have to use to join. All good? Okay, so that's just logistics. Yeah, okay, so PowerPoint slides. So let me just give you the big picture. So we ended up, and we're done with this data set now. We finished about a month ago, and it took about a year to collect. We're calling the natural scenes data set. And so in one sentence, it's a very large visual-oriented fMRI data set at Highfield on 8 subjects, and they came about 45 times scan sessions, which is kind of ridiculous. And we have a tiny little website here. It's just a placeholder, really. The data is not released. And the point of this workshop, I think, this is what we said in the little abstract. It's an intensive introduction to cognitive neuroimaging from a data science perspective. Okay, so this clicker, I don't know. So I mean, when I was figuring out what to do in these 5 days, I mean, it's very difficult because you can optimize for certain things. It kind of depends on the audience, what their expertise are. Kind of depends on what types of things you want to go over. And I ended up with this kind of design in mind. So the kind of analyses we do in my lab are, and I guess to some degree, many labs are on the continuum of this, of essentially programming your own analyses as opposed to using some software package where you essentially just click. And so definitely the nature of what's been designed is far on the side of program it yourself kind of from scratch. Although from scratch is always, it's not really from scratch, it's somewhere in, but there's a continuum along that spectrum also. But for what it's worth, it is very heavy on programming. That being said, I mean, these example scripts that exist, I mean, you didn't have to program, they're already there. And so you can theoretically still learn just by running each line and just looking at what's happening. But if you are adept with programming, feel free to just exponentiate the code and just go all out on trying to learn as much as you can. And I'm very happy, especially in the afternoon, where there will be some instruction where you'll just sit and watch my computer screen as I work through code, but I'm hoping the bulk of it will be, you will struggle with the code yourself. And if you're a good programmer, we can chat and I can show you stuff and you can show me stuff. Okay, so that's kind of the flavor here. And What we're also trying to do it's quite challenging, I think, but we'll do our best, is to try to encapsulate all the levels that you might understand how analyses work. And so this data set is huge, and if we're definitely taking a programming focus, there's challenges all over. So I kind of think of this at 3 different levels. You have to first, in terms of an analysis, to mine some brain neuroimaging data set, you need to know conceptually what you're trying to do in terms of the neuroscience theory and ways of thinking about brain function or structure. And you also need to know kind of the machine learning algorithms or regression, kind of the statistical concepts that we have to use given the type of data we acquire. And finally, you also, if you do take a programming approach, have to understand the coding level. And you can have expertise at any of these levels and might skimp on the others, but theoretically, you would want to have expertise at all levels. And that's kind of the goal here. I don't know if it'll come across as teaching that, but I'm going to try to do that. We're going to need another battery or something, or I'll stand over here. Okay, so this mainly applies to the example scripts that I designed. So I wanted to keep them as simple as possible while still not making them trivial. And that's a fine line to balance. Another principle, I think, is, I mean, and when choosing the content of what to cover in these scripts, there's a lot of different analyses one can do, a lot of different concepts. And in some sense, I kind of find value in many different ways of thinking about the data, or even thinking about the brain. So there's this multiplicity principle of many different types of analyses are useful, and all analyses have limitations. And I think maybe there's more personal philosophy, being cognizant of that and not like keeping your blinders on and actually thinking broadly about what you can do with neuroimaging data, because the possibilities are endless. I think that's a good thing. And so that's another principle that I was using when I decided how to design these example scripts. In contrast, you may hear people like, oh, I only do this type of analysis, or I only use this software package, which I guess I don't agree with. And finally, it is all in MATLAB. I think that was mentioned. Some of you indicated on the survey that you use Python, but for what it's worth, they're all in MATLAB. And moreover, we're going to use tools in the sense of functions that have been written. Again, striking a balance between, I mean, you have to use functions because otherwise your code will become 2,000 lines of code, and that's unwieldy. And we're going to be using these functions that we use in my lab. I don't think they're necessarily, like, I don't want you to lock you into these particular functions, and I think you will learn general principles as you go, even if you don't plan to use them in the future. And so I've tried to sort of optimize for generality also. Everyone good so far? Also, what we want to do in this workshop is to make it interactive, and I'm going to force some interaction very soon, so watch out. Okay, so this is kind of how we've designed things, and I think what you will get, hopefully, from the next couple days is to learn maybe types of analyses you're not familiar with. Also, how to deal with this very challenging issue. So again, this NSD, or natural scenes data set, is really, 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 really big, and that causes headaches. So on the one hand, you know, everyone wants more data and better statistical power and yada, 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 but it comes at a cost, and how to deal with that computationally is challenging, and I'm going to try to convey some of that as we go. And the last kind of learning point here is, I think by going through the example script and really thinking about them and reading them and seeing how they're structured, hopefully they serve as nice examples of good coding style. And I think I'll spend part of a lecture on going at least into some depth as to what I think that means. Okay. Oh, one last bullet point. Again, the kind of principle of multiplicity. A lot of it's programming, but I do use tools, ITK Snap. I use one or a few tools from like FSL, also FreeSurfer I rely on. So it's not like I only program my own stuff, but it's a kind of a smorgasbord, and I think that's okay, and it's good. I mean, if the tool is correct and you know how to use it well, then you might as well use it. And so it's not restricted to one package, but it kind of pulls from a lot of different sources. Okay, so that's all what it should be, and let me just get it out of the way. Like, importantly, you should know what it's not. There was a lot of kind of IT setup, so installing software on all these machines, getting the data downloaded and organized, and setting up this crazy setup of recording. Software, dealing with software is not what we're supposed to be learning, at least that's not what I was planning to go over. Also, the data have been pre-processed, and you might imagine with essentially terabytes of raw data coming in, you have to do the minimum stuff like motion correction, and that takes a lot of computational time, and that's not what the point of this workshop is, because that's more or less well accepted how to do this. Different programs, of course, exist, but that's somewhat boring, and we're not really going to go over that. Of course, if you have a specific interest in how we approached it or general conceptual issues and pre-processing, I'd be more than happy to discuss, but that's not what I planned the lectures on. And as I kind of alluded to, this is not an AFNI workshop or a SPM workshop. It's not specific to any software. And finally, in programming, you could spend a whole week or month learning programming, but we're not really going to formally teach it. That being said, I'm very happy to help you learn programming in the afternoon sessions if that's what you want to talk about more. Any questions? Okay, so these example scripts, I kind of mentioned this. It's a tricky balance, but I designed them to be as, I don't want them overly long. I also don't want them overly short. And they're commented a lot. So I hope you will take the time to read and execute the line and read and look at the outputs and like go through the entire script. And as I mentioned, I think they're examples of good coding, and you can kind of see if you agree. And if you do agree, then you might use them as a model for how to structure your next programming assignment. So these examples, as I mentioned, there's 12. Actually, Boss has 2 more on diffusion, which I was not aware of until this morning. But these scripts happen to be on this GitHub repository that you see right there. And I also took the scripts and used MATLAB's publish capability. Publish is like a function that essentially takes a set of code, runs it, and then generates these little web pages. And so that also exists if you just want to browse what it should look like when you run it. And you can just go there, and there's a little link, and just click on it. Then you can just essentially read the script and look at all the outputs without actually running the script. And anyway, so over the course of the next 5 days, we will be going through each of these 12. Okay, I think hopefully this is of interest. I did want to mention this slide, sorry, the next slide. Basically how things are set up. It took a lot of kind of thinking of, as you know, neuroimaging has many software packages, but getting everything set up on these iMacs took some thought, and there's some software that I use that may be of interest to you, and I just want to go over the ones that I've installed. So we can think of what we've prepared in terms of data. It's actually split into data, software, and code. So the data, by data I mean these gigabytes, terabytes of files after pre-processing. So I'm not giving you raw nifties or raw DICOMs. These are after basic things like motion correction and registration. The data kind of are static. We don't want to touch them. You will have to trust that they're done well. And so that's distinct from what you might call software. So this includes FreeSurf FSL, and there's a whole slide on this in a second. And then the third distinct bit here is the code. And so the code will undergo rapid development. You're not going to touch the data. You're not going to touch the software. They're just executables. But it's really the code, these scripts, and associated toolboxes that you will be focusing your attention on. So I've kind of segregated the materials into these kind of categories. And as I said, I want to just briefly, I don't want to spend too long on this, talk about the software. So let me just skip over this. You will see this in the afternoon. You will see where the data are. It's mounted. It's on a network mount because each computer had. I mean, this is terabytes of data, so we can't stick it locally in every iMac. So it's mounted on HPC, the system you guys have here, I believe. And so you will see that in the afternoon. Maybe I just need a new battery. I'll do it later. I'll just do this. Okay. Yeah, so the software. So back in the day, a couple months ago, I was deciding, well, what software will I use? I didn't even know what kind of example scripts I wanted to show. But I ended up installing, with the help of Osama, who's not here, a couple things. So OS is pretty boring. These are all Macs. So I installed basic OS stuff. In terms of neuroimaging, there's FreeSurfer on the computers. I guess we will use FreeView, which is the surface viewer component of FreeSurfer. ITK Snap is one of my favorites. We'll be using that a lot. It's a very useful volume-based viewer. SPM, everyone's heard of. We're not really going to use it, but it's there. FSL is on the computers, AFNI. And MRI Crow, also a volume viewer. I used to like it, but then ITK Snap, I feel, is even better. So I switched to ITK Snap. Anyway, so these are all useful, and some of you may not have heard of one or two of them. You might want to check them out. Things that might be called just programming. So MATLAB is on all the computers. There's also Psych Toolbox. I'm sure some of you use it. Turns out we're not doing an experiment, so we don't need Psych Toolbox, but it is there, and I use it to run experiments. And Python's, I believe, is on the computers, although I haven't really stress tested it. So if someone is interested in actually doing Python coding on the prepared data, I'd say go for it, and maybe you can teach me how to use it. Okay, so that's all on the computers, and a bunch of utilities. Zoom, I find very useful. We're using Zoom right now. It's a replacement for Skype. FileZilla is just a file transfer utility. And then it's some basic kind of office productivity stuff. I like BBEdit. It's a text editor. It's only for Macs though. So if you use Windows, you must use something else. Okay. Oh, and video stuff. So some of the example scripts will actually use some video processing. Videos are just very handy figures. It's essentially many figures in a row that you can watch, and it's very useful. And just getting familiar with how to generate these videos from code or software and then manipulating them can be very useful. And so I have installed some basic software. Handbrake and FFmpeg are kind of video transcoder utilities. They're all free. And ImageMagick is not a video. It's image for images, but it can do conversion. A lot of these, I mean, the goal here is like, yeah, you can do it by hand in your favorite photo editor, but automation can be very useful. And so ImageMagick comes with command line utilities that you can then put into a for loop and quickly process a lot of images. Okay. Any questions or comments on software? Okay. Maybe I'll do this. Just a very brief intro, and then we'll go hands-on in the afternoon. We have here essentially what you should see on these iMacs. So there's a lot of clutter, but let me just highlight what you will be looking for and just get your head on straight regarding what we have set up. So this is the network drive called Vision. And inside of Vision, on the window to the left, we have a couple folders. You can ignore some of those other folders, I think. Maybe diffusion will become relevant on the last day. But this folder called NSD, as you can see there, that's the large prepared data folder, terabytes of data. So that's going to be of interest. And specifically, the NSD data folder inside of that is the primary folder of interest. So just kind of a forewarning of what to kind of zoom in on. And then the one other component of interest here is on the desktop of each computer, there's a NSD Abu Dhabi folder. This contains the code. So remember what I deem code, not the software, not the data, but just the scripts and stuff. And so this folder is shown here, and it contains the lecture slides, also some useful documents. The main ones that will be of interest are these 12 text files. And it's just a bunch of text, but that's what we'll be going over in the afternoon. Okay. So we did take a survey, and you guys, I'm not sure it affected how I designed stuff, but I was curious to know what people's backgrounds were. And let's see. So this is from the Google Sheets. This is the survey results. As you can see, blue is for people who profess to have no experience. red is some experience, orange is a lot, and green are the professed experts. And I think the take home is there is a spread. There's a very large spread. There's people that fall in all the bins on these various topics: cognitive neuroscience, vision. Oh, memory. I call it a visual data set, but the experiment actually involves a memory component. So you could take the data and answer questions about how memory works. So that's why I added that question there. Uh-oh, you got to click on the thing, yeah. Okay, I also asked about what's your familiarity with Linux, using things from the command line, MRI data, MATLAB, Python. Again, pretty large spread. I guess no one's an expert in MRI surface format. So that's good, because that's some of what we'll cover in this workshop. It's good that everyone has at least some experience in MATLAB. There's no blue there. I also asked questions about neuroimaging packages, FSL, AFNI, FreeSurfer. Ironically, almost no one has used ITK Snap before, but that's good because that's what we'll go over this afternoon. It looks the winner out of neuroimaging is FreeSurfer. It's the distribution that has most mass towards the right. I don't know, take it for what it's worth. I also asked questions about like analysis and statistics. And again, large spread. I'm not sure what to do about this, which just makes it difficult because you may be bored if you already know the stuff, and some of you who are new to it might not. So we'll just see how it goes. And then lastly, kind of neuroimaging styles of analysis, GLM analyses, encoding models, MVPA, functional connectivity. I guess those distributions are quite leftward masked. So the mode of those distributions is on no experience. So that seems a bit daunting here, but we'll do our best to kind of convey what we can. Okay.
Do you think it's now a good time to go around the room and get like everybody their background and what they hope to get out of the workshop?
That's a great idea, but let's do it this way. Go in groups of 3, and I want to know, and we don't want to spend too long on this, but I do want to know, either tell me a topic that you are generally interested in outside of this workshop, just like stuff, either a topic in neuroscience or an analysis approach that you seem to have some interest in, either that or something you are hoping to get out of this workshop. So pick one, discuss in groups of 3, and then we'll reconvene in like 5 minutes. Yeah, and I had a few thoughts I just wanted to share based on what I heard. They're kind of random thoughts. So one thought is, don't listen to what I say. Like in some sense, this is my own, a lot of this is my own kind of take on things. So I think you should pass it through a skeptical filter and think about it and argue. Also, it's not like I know everything about all possible topics and analysis, and especially not neuroscience topics. So it's a bit embarrassing to think that I can. So I heard a lot of interest in connectivity, and I mean, I know of it, and I do a little bit of it, and I have opinions about it, but don't take my opinion as ground truth. And also, MVPA encoding, all these things, I do those a lot more, and I have opinions, and I'll tell you what I think and justify it, and you can decide if you agree or not. Other random thoughts I had. So yeah, this NSD thing, I mean, I'm sure some of you heard of, say, the HCP effort. So the Human Connectome project happened at Minnesota. I had no official role in it. I mean, I was semi-officially helping out with one part of it. But I guess this data set we have, the NSD data set, I mean, it's similar and different. It's similar in the sense that it's going to be a publicly available, hopefully widely used data set of fMRI and MRI related other types of MRI, not just fMRI. So it's going to be of that ilk, I guess. But one major difference is, we have 8 subjects longitudinally scanned many, many times, whereas in HCP, it's more of a model of many people, over 1,000 scanned, not briefly, but not super extensively either. So you can kind of mentally group NSD a little bit in that realm of large scale, publicly available neuroimaging data sets. Also, I heard a bunch of resting state interest. So we did, and Emily's going to tell you more about exactly what we did, but resting state was included, and we took it very seriously. Sorry, I lost my train of thought. Oh, right. So it's there. It's actually on these machines, as well as on the server, and it's now accessible from these computers. So you could choose to do a little project on actually tackling the resting state. I mean, there is an example script on it, but it's very keep it simple, stupid. So it doesn't go very deep into it. But the data do exist, and it is accessible on the server. So we could talk more as to what specific things you might want to look at in the resting state part of the data set. Other thoughts that I got. Oh, MEG. So John Winnower might be doing the NSD MEG component, meaning it's not going to be the same subjects, but it'll be different people, which of course puts limitations on how much you can mine from this, but similar spirit. Recruit really good subjects, scan them a lot, and use the exact same experiment or something as close as possible so the stimuli are matched and things like that. And if he does it, and if he makes it publicly available, it will be out there. I don't have that much experience with EEG or MEG, and I have a little experience with ECOG, so I can't really speak to the technical issues there. But certainly, as has been mentioned, like the analysis techniques are general. Like time is a different order of magnitude, seconds versus milliseconds, but in terms of programming and analysis, it's all the same stuff. So I do think it should transfer. Sorry, my gears are slow today. I think that covers it. Okay, so we're doing okay on time. We do have 3 lectures, but the second one is medium scale and the third one is really long. I just have one last set of slides here. It's going to be me on my soapbox. Again, you don't have to agree with what I say, but this is where I come from. This is how I design these examples and why I do the type of research I do. A lot of this, actually, the bulk of this workshop is really on methods. I mean, certainly there are cognitive neuroscience topics hidden here, vision, and I study vision in particular, and so we can talk about vision, but it's not really about vision, this workshop. It's about analyses and coding and statistics. And because we're taking a very do-it-yourself approach, so it's not like, let's learn how to use software package X, it's more like, what the heck are they doing and what operations are they performing and how do we actually program those operations if we want to do something different than what's been already coded for us. So here's my kind of philosophical take on these matters. And I actually made these slides, didn't I give this at a lab meeting? And I got a lot of pushback, but well, see what you think. So there's 2 kind of extreme attitudes that one might take. Maybe you can drag that window over. So one attitude is kind of, don't reinvent the wheel, use the things out there. I don't really care about what they're doing under the hood because I'm busy, I gotta get papers written, I just want results, right? That's attitude 1. And it's kind of an optimistic attitude of like, what's out there, tons of people use it, it's probably fine, right? Whereas the other attitude is very paranoid. It's like, I don't trust that thing out there, and you test it and you look that it fails in certain cases and you really want to know what it's doing and how to make it better. It's probably obvious which side of this continuum I'm on, but I want to be fair. Like, everything comes at a cost and everything has certain purposes. And so I think my take on this is you have to be judicious in when you apply each attitude. So according to me, when is it okay to kind of take the lazy attitude and when is it okay to take the more time-consuming attitude? So my view on this is the following. I'll just read it to you. It kind of depends on the situation. So insofar that you might meet these 3 conditions, 1, 2, and 3. So being lazy is fine if the thing you're using that you don't really understand is like foolproof. Like, there's only one way to use this tool, this black box. Condition 2 is there's only really one like it's very constrained. It has like a single purpose. One input, you know the input. It's very simple. It's not like 20 inputs. You just feed it one thing and you get one thing out. So the more it's like that, the easier and better. And then the third condition is checking. So things can go wrong. I'm sure many of you have experienced this yourself, of like you have some large data and you pump it through something and like you look at the output and it crashed or it doesn't look right. Anything can happen and often things will fail. And so it would be nice if you can verify in a glance, sanity check, quality control, all of these words, all these phrases kind of mean the same thing. Can you check the results of the black box very quickly and effectively? So if all these three conditions are satisfied, then great, let's be lazy and just use the thing as is and move on. Or at least this is me talking. Now, if any of these conditions fail, or if all three fail, then you might want to not take attitude 1. So let me give some examples of when it's okay. So I think free surf is a very good example of a good thing. Like you feed it a T1, a 3-dimensional anatomical volume, typically at 1 millimeter. And you can, before you run it, you can at least eyeball the T1 quickly, and it looks like a brain. You see white matter, gray matter, it looks reasonable quality. Okay, great. You feed it in, gigantic black box turns. Typically, it takes a whole day to run. And then you just go off and do other things and drink coffee or whatever. And then you come back and you just look at the surfaces. So I use it primarily just to get the cortical surface reconstructions. It does have many other outputs like segmentations and all this stuff, but I really just care about the surfaces. And it doesn't take that much time to look at the surfaces. And what you see is what you get. You can just see it. You know it's correct, unless there's some insidious problem with the viewer. But so I think that satisfies these conditions. There's really, in the simplest case of using free surfer, there's just one way to use it. Recon all t1.nai.gz, done. And then it goes off and does its thing. I don't really care what it's doing. It's doing magical stuff. But I can take the outputs and be like, and look at it. And in 1 out of 20 times it might fail. And then you have to worry and bang your head against why is it messing up, missing an entire part of the brain. But most of the time it seems to work. And you can verify that. So that's my favorite example of a good tool. Like it's single purpose and you can verify its results and it just works. And then in that case, yeah, I'm not gonna write my own free surfer. Okay, so that's the example in neuroimaging. And then, many things we do outside of research is like that. My car turns on, I drive it. I don't know how a car works, nor do I care. Light switch, I flip my finger and light occurs, right? And actually, I don't really know how electricity works either. So you could say like, yeah, I'm taking attitude 1. Google Maps, we use it all the time and it just works. And the algorithms that they use to determine the shortest path, all these computer sciencey things, like, yeah, you could know that, but like that's not the point. The point is, I want to know where I want to go, tell me, and then it works, right? So these are all like examples of attitude 1. But if any of these conditions don't work, so if there's multiple ways to use it and many finicky inputs and parameters, and we don't really know what the right answer is, if any of those conditions hold, then I would argue we need to worry about attitude 1. And my example of that is fMRI. In general, so I mean fMRI in the big picture. So take this several runs of data from a subject with an experimental paradigm and like go all the way from start to finish. I would think, and maybe you agree, although I want to see what you guys think, I think that conforms to essentially failing all 3 conditions. It's hard to confirm that it worked. Like, what is the right answer? We all hope to find something that looks good, but that's not really a criterion. And there are many analyses one can run, many different software to run. So all of these conditions, I think, fail. And so maybe that's motivation to want to peer under the hood, think about the basic statistical operations that software packages implement, and actually verify them and look at them and think about if they're doing doing them correctly. So any gripes or agreement or disagreement or comments? I see some facial expressions that are expressing gripes. Oh, no, I agree 100%. So sorry, that's what I meant by in general. I meant like the whole thing. But like if there's a very prescribed task, like get this brain to be matched to this other brain, motion correction or registration, that is very almost, yeah, very clear cut. And for that specific step, I agree, that could be thought of as I don't care how it works, just do it. Okay, a little bit further, and then I just want to, yeah, I should speed up. So these are different ways that I think about methods. So this we've already talked about, the black box that you have no idea how it works versus opening it up, and then you're confronted with immense complexity, and then you get overwhelmed. Neither is great, and so this is a struggle that I think we all have to deal with. In terms of like software and tools, often you will use something you get from someone else, like software packages or like a function that someone writes for you. And so you have to think about the roles that you might play. So you might be the user of the tool, and if you're under that mindset, there are certain things you want the method to satisfy. You want it to be easy to use, clear, easy to install. Whereas, you know, you can put on your developer hat. So insofar that you write code that you run yourself or you might give to someone, then you're occupying a different role, which is you're designing, you're writing the documentation of the code, you're testing it hopefully before you unleash it upon someone else. And so, you know, these roles are, I play both hats, and like sometimes I develop and then I switch gears and I'm like, I'm the user, and then I look at the code and I'm either happy that I developed it well, or I have to go back to be the developer and then fix it, right? And again, you know, maybe you don't want to be a developer. Maybe you've decided you will never be an expert programmer, so you will never, you know, write a function for general use. That's okay, but you know, but you just have to decide for yourself who you want to be. Other metaphors we kind of went through is, I don't know how a car works, and that's okay because cars are well designed and they work. Maybe unlike some fMRI analyses. And then last metaphor here is, this is kind of the neuroimaging package metaphor, which is, how do you cook? Well, you take this thing and just put it in the microwave. That's like using some pre-canned GUI or something, versus do you know how to actually cook and put the ingredients together and do stuff? Again, I'm not saying you necessarily have to take attitude 1 or attitude 2, but you should at least think about it explicitly. Okay, last slide. And this is me going off the deep end. I would say ideally, I mean, this goes back to the data. So fMRI data, why is it so complicated? Well, it's because you have so many voxels and so many time points and so many runs and so many subjects. And it's not just fMRI data, you have to deal with structural data and behavioral data. And so there's just a lot. And all those numbers, my kind of metaphor is like, these numbers are getting transformed and combining with one another, and then the end product really is a figure in a paper. And so at the end of the day, it might just be like a 1 bit of information. So like the height of a bar, or the brightness of a voxel in a slice, or something like that. And so that's massive data converging onto this like one magical number. And so that's really complex, because hundreds of thousands of numbers kind of commingled together to generate what you hope is an accurate reporting of a result. And it's complicated. And so the ideal, I think, is it would be nice if we could check all of those transformations. And that's very hard because there's too many numbers. We can't check it by hand. And so what we have to resort to, and this is kind of foreshadowing some stuff later on, is you either write code and test it with simulations, or you write code and verify the correctness using your head, or you write code and verify it using, you know, benchmark data sets or something. And none of this really happens a lot out there, I think. So that's unfortunate. And so confirming correctness, I think, is hard and not really done a lot. In fact, there was a preprint by Brian Wendell actually, on this. He, in case you're interested, PRF methods, he and I think postdoc working with him just wrote a paper on just taking software packages, one of which was mine, and essentially comparing them on a ground truth data set. Like, very kind of principled, simple thing, but like, again, that's like the exception. Anywho, so let me just skip this a little bit. So the ideal is you would understand everything that happens to all your data, and that you would describe this extremely clearly in a paper and so on. And I would say this is ideal to strive for, if possible. And one last thing on specific to NSD, we wanted to create a huge data set, but that's going to make all of these things even harder. For two reasons, I would say the largeness of the data, and the more sophisticated the analysis you want to apply, the largeness of the analysis. So people here said like, oh, I'm interested in like machine learning and whatever, and that's great, but these are more complicated analyses. So not only are the data huge and complicated to wade through and complicated to convey, also the analyses, as we become more sophisticated data analysts, become also complicated. So now you're hit doubly. Again, I don't have a solution, I'm just pointing out we should be cognizant of this. And so this point is, we hope we do a good job conveying what the data were, what the experiment was, how we prepare all these files, and clarity and someone describing what they did is like kind of the precursor to actually doing good analyses. If they document it poorly, then you don't really know what they did and you don't really know what the subjects were doing, and that's just not a good situation. So yeah, so in general, I like simple analyses. So certainly I do complex analyses, but I'm not doing them just to be complex. I'm doing them because they're necessary to kind of solve the scientific questions that I'm trying to solve. But if you have a choice between two analyses, one is simple and gets the job done and is interpretable, and one is complex and convoluted and has all these weird choices, I would choose the simple one. So I don't know if you agree, but that's my take on this. And let me just end with this last bit. Nowadays, people are about open science and sharing data and code and so forth, and I generally agree with that. But I did want to point out that it's not just sharing code. Just by sharing your code doesn't mean that it's correct. And I think we also need, in addition to sharing the code, to describe it clearly. So this means like code comments, and also how you write your method section in the paper is really important, because it takes a lot of time to look at someone else's code, and basically it doesn't happen. We're all busy, we have our own projects, and unless like a group of people become very interested in one paper, it does not happen. So just by putting the code out in the open doesn't, I mean, it's nice, I'm not saying don't do it, and I do it, but it's not like, that's not the only thing to do. So developing clarity in your head at the conceptual level. So remember, I just said, you know, there's the conceptual level, the algorithmic level, and then the code level. I mean, you kind of want all three. So sharing your code helps the third level. And people can look at the code, and you can look at the code and at least check the code. But describing the operations that the code implements is also really important. And that's kind of the method section of your paper. And I personally spent a lot of time doing the best I can to describe in prose, English text, the operations, because no one really wants to look at code. People just want to understand the algorithmic level, the statistical operations that you perform. Okay, that was it on my kind of sandbox, soapbox. Did anyone have any thoughts? I'll let this churn a little. I mean, the reason I'm telling you this is it's kind of what was put into these example scripts and like the design of the lectures and so forth. And so this is where this is my stance, and this is why you'll be hearing what you will hear in the next few days. Okay, quick poll. If you had to choose one, attitude 1 or attitude 2, which one do you generally take? Let's see a raise of hands for attitude 1. Don't be shy. Okay, let's see raise of hands for attitude 2. Oh, it's pretty mixed. Okay, interesting. All right, let's take a little break, and then like just a few minutes, 5 minutes, go to bathroom break or something, and then we'll dive into the second of 3.